Hi everyone, nice to see you. I hope you're doing well. Thank you so much for staying for lunchtime. We know that usually food is much more important than science. So thanks a lot for staying and being here to attend this session. So the talk that you're all waiting for from Dr. James Phil will be in 3 minutes. Before that, I just wanted to give you a brief introduction about GenScript and myself. So I'm Linda and I'm actually French. I've been with GenScript for about 5 years. And I actually used to work in UK in antibody and protein engineering, so it's really like one of my key loves in my life, let's say. I was in UCB Pharma in Slough, and also in Oxford University, working on spike catcher technology, which we can see in quite a few things now. But you know, it was UK, bad weather, bad food, bad wine. I had to go back to France, kind of. So that's why I went back to France, where I'm located near Paris. And to tell you a bit more about GenScript, so we are a CRO that actually supports scientific in 5 main segments: cell therapy, gene therapy, IVD, vaccines, and today's focus is going to be on ADD. Marianne? Sorry. Is there a pointer or? Oh, I'm on the wrong side. Sorry. Okay. No. Yeah? Good enough? Okay, cool. So, and to go back, it's the red one? That perfect. Works well. Okay. So, today, all of you guys in the room are involved in antibody drug discovery in some way. And I think that we'll all agree on the fact that in your daily job, everything you do is to really have an impact and treat a disease and support patients' health and life. To do so, you're going to need a strategic approach, which James is going to talk about quite a lot during the presentation. You need high quality, you need speed, and you need trusted partners that are going to support you in the journey. So, how does GenScript support ADD? We will be able to talk more about that at booth 1218, so please feel free to come, because today we don't want to take too much time talking about, let's say, what we are doing. We really wanted LabGenius and James to really talk about their technologies and the cool things that they are doing. But the take-home message on the GenScript side is that our role is really to support scientists worldwide by helping you at every single step of the way, from target identification up to clinical development. And to do so, we've got a wide range, again, of services, products that we can discuss later on. But before James can start talking about his presentation, the one thing that I still wanted to highlight before we can dive into the talk is really the product or the molecule that you are going to need at every single step of the way, which is antibody, in antibody drug discovery, right? And for antibody, the one thing that I wanted to emphasize here is really recombinant antibody production, and a specific platform that I still wanted to give you a brief introduction about, because James will talk about it just a bit. Again, we don't want to focus on that. But here, since in the room, there are many people working with many different type of formats, full length, bispecific, trispecific, different type of fragments. That's why we wanted to give you an introduction about the TurboCho platform. TurboCho, the name is extremely clear. I actually kind of like the name, you know, cool, Marvel, very, very clear, TurboCho. It's really a game changer in the recombinant antibody production market. So you have GenScript R&D and bioinformatics team, that join forces and combine our best technologies into one single platform. When I say best technologies, that is codon optimization, a very powerful one to maximize the yield. That's better media in order to be able to express in a very effective way. That's also very high quality processes to make sure that you can get very good molecules even after one-step purification. And basically, you can wonder why did we decide to actually focus on a CHO platform instead of a HEG one? Because many people in labs still work with HEG cells. So as you know, most of the commercial antibodies of the market, actually all of them, are CHO-based. So we really wanted to have a platform that you can kind of use from start to finish. And TurboCho will help you to actually get small-scale production, like microgram level when you're doing small-scale production. That's something that James will talk about. And with this platform, you can actually go up to gram and kilogram production. Here, you might wonder a few things about TurboCho. So why is it called TurboCho? Because it's super fast. So your question might be, okay, I'm sure it's not your question, but let's play the game, okay? So is it possible to produce recombinant antibodies in 5 business days only? Yes. Is it possible to get yields up to 3 grams per liter in a transient way? Yes. Questions are getting longer and longer. Is it possible to obtain up to 95% purity, just like HPLC, For bispecific and other complex formats after only one step purification, yes. Is it possible to use TurboCHO at a similar or lower cost than actually in-house production? Because well, money is also like a key player, right, in the market of HD. Yes, it is. So basically, am I saying that you can obtain fast production, high yield, high quality, and cost effectiveness with a single platform? Yes, that's the TurboCHO platform. So again, James will talk slightly about it, just the part that they're doing with us, but since there are many researchers here that are working on different type of formats, you might need other things, so that's why I wanted to give an introduction. But speaking of James, let's move to the importance of AI in ADD field and LabGenius mission. You might all know this cycle, which is the AI cycle. So for AI drug discovery, basically, I wouldn't say it starts because it's really a cycle, right? There's no real starting point, but let's start with design. So kind of start with the design, in silico antibody design. Then you build with high-throughput protein production. That's something that we have discussed before with TurboCHO. Then you test by doing validation by high-throughput functional and developability assays. And then you're going to build and refine the machine learning models. Then you start all over again. So what's LabGenius' role in that? LabGenius is pioneering the discovery of complex therapeutic antibodies by combining machine learning with high-throughput experimentation. And Dr. James Phil, CEO of LabGenius, here today, will be able to tell you more during his talk, which is engineering T-cell engagers with complete killing selectivity through the closed-loop integration of machine learning and high-throughput experimentation. Now you might wonder, once again, who is Dr. James Phil? Dr. James Phil has a PhD from Imperial College. He did his PhD there. He raised more than 17 million in financing from top-tier venture capitalists. And also today, through LabGenius, there are many deals and many partnerships with biotechs and top pharmas like Sanofi using their technologies to bring the next generation molecules. So thank you everyone for your attention. And now I leave the stage to Dr. James Phil.
Thanks so much, Linda, for that introduction. And thanks again for everybody for missing their lunch to be here. I really do appreciate it. Okay, let's see. Okay, yeah, great. Okay, so as Linda said, our mission is to combine machine learning with high-throughput experimentation to accelerate the discovery of novel therapeutic antibodies. And what I'm going to do today in this presentation is talk a little bit about the work that we're doing in the world of T-cell engagers to increase the selectivity of these molecules. Now, for those of you who haven't heard of LabGenius before, we're a London-based company. Let me see if this is going to, yeah, very good. We're a London-based company we're staffed by a team of 60 very talented scientists and engineers who, over the last few years, have really brought together a closed-loop platform that is allowing us to discover novel therapeutic antibodies. And we're doing that both in the context of our own internal pipeline, but also in collaboration with our pharma partners as well. So as I say, what I wanted to talk to you today is around how we're deploying this approach in the context of solid tumors. And I've heard in so many of the presentations over the last day and a half that a challenge that we all face is how do we get our antibodies to go to the right place and have the right action. And this is a particularly acute problem if you're working in the world of solid tumors where, quite frankly, we just don't have enough tumor-specific antigens. Now, obviously, what we do have is a boatload of tumor-associated antigens, which are, of course, expressed on both healthy and disease cells. But if we're going to go after those, the big challenge that we face is how can we go after them and overcome this challenge of on-target, off-tumor toxicity. Now, for those of you who are sort of maybe unfamiliar with on-target, off-tumor tox, think of a first-generation CD3-based T-cell engager, probably high affinity for that tumor-associated antigen. They indiscriminately bind to that antigen, be it present on healthy or disease cells. You get the recruitment of your T-cell and the indiscriminate killing of both your healthy and disease cells. And this has been massively problematic for that first generation of T-cell engagers. Either they never even made it into clinical trials, or they did get to clinical trials and they showed promising efficacy, but at a level of toxicity that precluded them from being taken forward. Or maybe where we did have some approvals is the patients who have to deal with those with those side effects. And what it really comes down to, for me, is these curves that you see across sort of every paper that you look at, which is looking at how these molecules act on both healthy and diseased cells. And of course, what we're really interested in doing is widening that window between the activity of our T-cell engager on the healthy cells and the diseased cells. And of course, we've got all sorts of tools in our armory now that allow us to do this, whether you're looking at pH-sensitive binding, whether you're looking at masking, whether you're looking at avidity-driven methods. These are all ways in which we can widen that therapeutic window. But in the majority of instances, it is just that. It's just widening a window. And the question that we've been asking ourselves is it possible to go beyond widening that window? Is it possible to actually increase it so that we have something approaching complete selectivity? There we go. Yeah. So this is the question, the open question for us. To what extent, irrespective of the different technologies that you're using, pH-sensitive binding, masking, avidity-driven mechanisms, can we achieve something that's approaching complete on-off killing selectivity so that we can then go after TAAs that are found on all of these solid cancer cell lines? And in order to do that, effectively what we need is molecules that are able to go beyond just binding to a single target. We need them to be able to effectively recognize multiple targets simultaneously, be them the same TAA or different TAAs, and sample the surface of the disease cell and effectively make a decision over which cell to kill or not. That is the challenge that we're looking at. And at LabGenius, our hypothesis is irrespective of the target of the selectivity mechanism that you're using, those molecules with that incredibly high intrinsic selectivity exist, but they're just really challenging to find. And I want to take a moment to talk about why they're challenging to find and how we might be able to overcome that challenge. The first piece is when you characterize any of these complex therapeutic antibodies, you have to go beyond just binding. What you really want to understand is how do these molecules work in the context of really disease-relevant complex cell-based assays. And historically, those take time to set up, they take time to run. That limits the number of variants that you can explore. But the approach that we're taking at LabGenius is to say, what happens if we invest a lot in the robotic automation behind those complex disease-relevant cell-based assays and then layer on machine learning? Perhaps we can then open up the design space that we can explore and actually evaluate a very large number of these different T-cell engagers and potentially find ones with really exciting selectivity profiles. So that's exactly what we've done. Brought together those two pieces, the high-throughput experimentation and the machine learning. And what that's enabled the team to do is explore design spaces up to 10 to the 6, so a million different antibody variants, where you can then evaluate those molecules, both for function and for developability. And I'll talk a little bit more in this presentation around how we do that and the results that we've obtained with that specific method. Now, I apologize for the fact this slide has a lot of words on. Please bear with me. The actual approach that we take is remarkably simple. So the scientific team will demarcate that area of design space that they want to explore, and that will exist in silico. Now, we could never experimentally test 10 to the 6, a million different antibodies in the context of these disease-relevant, complex, multi-day cell-based assays. But what we can do with a lot of high-throughput automation is to subsample that space so we can take thousands of those molecules and then experimentally characterize them. And use that characterization data from both the functional assays and also the developability assays to build models that are predictive of each of those features. And then we can query those models to suggest new designs that we can then, again, validate in the lab to get high performance. And in the case of the work that we're doing at LabGenius, the specific mechanism that we're using to differentiate between healthy and diseased cells is an avidity-based mechanism. So we're trying to build into our antibodies the intelligence to be able to sample multiple different tumor-associated antigens simultaneously and then make a decision over which cell to kill or not. So we're looking at differential density of TAA expression as a primary mechanism to differentiate between healthy and diseased cells. And the antibody format that we've chosen really reflects that specific mechanism of action. So this is what one of our typical T-cell engagers will look like in terms of its architecture. The first thing you'll notice is you've got multiple different TAA binders chained together. That's really important. That buys you the ability to simultaneously sample different TAAs simultaneously. So shown in yellow, you've got your anti-TAA binders there. Now we join these together, and when we explore these design spaces, we're looking at loads of different linker lengths of different rigidities and flexibilities and lengths. And again, why that's really important is it buys us the ability to explore unique geometries, and we found that has a big impact on the performance of these molecules. You'll notice that we've got monovalent anti-CD3 binding. That is, of course, intentional, and that's built in so we don't get premature activation of the T cell prior to immune synapse formation. And an asymmetric FC on there. It's a silenced asymmetric FC for half-life extension. So what I'm going to show you now is just mechanistically how that avidity-driven process works. So we have a healthy cell here, low TAA expression, low affinity interactions of that TCE with it. Not a sustained interaction isn't formed, the TCE floats away. But when it encounters a disease cell where the TAA expression is much higher, that molecule is then able to come in and you have these multiple low affinity interactions actually resulting in a sustained binding to your disease cell, and then you can recruit in your T cell to get that targeted killing of the cancer. So this is really a very intuitive mechanism of action, but the challenge we then face in the lab is how do we co-optimize these molecules, not only for selectivity, but also for potency, for efficacy, and also developability. And how can we do that? I'm going to talk through the process that we use. So here are the different levers that we have when we're considering the design of these different molecules. And when you're searching a design space of many hundred thousands of these different antibodies, you've got to take each of these into account. So we're looking at the different linker lengths between the binding domains. What's the rigidity of those linkers like? The affinity to which they bind to your tumor-associated antigen. You bind too tightly, you stick to everything. You bind too loosely, you'll stick to nothing. The valency, so the number of different binders that you put together, which of course impacts developability. The topology, which we found to be really important in the performance of these molecules, so where you put the pieces relative to one another, and the epitope. Are you targeting a proximal or a distal epitope, which of course impacts immune synapse formation and has a big impact on the performance of these molecules. And the challenge for us as human protein engineers is it's really hard to draw a straight line between how you change these different levers and the performance of the molecule. And why is that? It's because each of these different factors there's a complex interplay between them. And not only that, you're then trying to co-optimize across multiple different features in parallel. So whilst this is a really challenging engineering problem for a human, it is the perfect problem for machine learning, contingent on your ability to generate enough high-quality machine learning grade data to train your models. So what I'll do now is just talk you through a case study where we've deployed this exact approach to the optimization of one of these T-cell engagers. And here we were looking at a tumor-associated antigen where there's only a threefold difference between the expression levels on the healthy and the disease cells. So just for context, that's actually a really small delta there. You're looking at something between, 7 to 10,000 receptors on a healthy cell, 25 to 30,000 receptors on a disease cell. That's a really big challenge for this antibody to tell the difference between those two cells. And I'll show you how we were able to achieve that. Okay, so the very first part of our process, this is the lead discovery part of our process. What we require is really diverse pools of binders for each of our subparts. Now, of course, we have our anti-CD3 pool that we can deploy across all of our different programs. But on a program-by-program basis, what we require is new anti-TAA binders. And specifically what we're looking at there is we need low affinity binders, high affinity binders, everything in between. We need diversity with respect to epitope coverage. And at the start of any program, we'll both screen phage libraries, both synthetic and naive, and also initiate immunizations and pool everything that we have together. Do all of the in silico work to optimize those binders. But the really key piece is how do we then go from having this very long list of sequences to a really nicely characterized pool of binders for this initial lead discovery part of our process. And that's where we've been able to build a really fruitful collaboration with the folks at GenScript, because what we've been able to do is ship them off hundreds and hundreds of sequences, and then in a 2-week period, they'll ship us back the protein for those mono VHHs. Really reliable, way. So we get very fast protein, it's very high quality, and the yields are great for what we require. Also, the purity of those molecules, we see really great percentage monomer typically across those different variants. So by working with partners like GenScript, what that enables us to do is to really do a lot of the heavy lifting on that early lead discovery part of the process, and allows my team to really focus on the lead optimization side, which I'll talk about now. So once we have all of those really nice pools of binders, we can then start the lead optimization process. Now that process starts with asking our scientists to come together, and this is a point where real scientific ingenuity is kind of injected into the process. Using all the information that they'll have, they will demark an area of design space that they want our closed loop system to explore. So in the case of this specific project, we had a pool of 25 TAAs across a couple of different epitopes, 3 anti-CD3s, both VHHs and SCFEs, and a library of different linkers. Valency of between 1 and 3 on the anti-TAA side. And when you look at the combinatorial mix of those, you've then got a design space of over 270,000 different molecules. Now, using brute force screening alone, there's no way that we can take all of those 270,000 molecules and characterize them, again, across these really in-depth, complex, disease-relevant cell-based assays. But what we can do is we can subsample from that space and take thousands of those variants and characterize them, again, across this whole battery of assays, use that data to inform, to either train or refine models that are predictive of the overarching space, and then query those models for high-performing sequences. We use that process very effectively in this program. Now, the heart of the LabGenius platform on the lead optimization side is, again, this design, build, test, learn loop that Linda mentioned previously. And I don't have time to go into a huge amount of detail, but I'll try and pull out a few key points that I think are relevant and might be of interest to this group. The first piece is we go from design, build, test, and learn. It's a 6-week process. The fastest we've done that is in 23 days. I don't think it's possible to get that process to be any shorter just on the amount of time that it takes us to grow cells and also to run these disease-relevant cell-based assays. So we're looking at several multi-day assays testing across multiple different PBMC donors, etc. In terms of the build stage, we have a mass modular cloning process that allows us to create these very large panels, over 700 different antibodies, roughly 768, and they get then sequence verified with in-house nanopore sequencing. All of those molecules are then expressed using a mammalian expression system, and that's really key. So we do all the screening in final format or near final format screening, obviously really key, all expressed in a mammalian system, which is important for the downstream developability assays. And then we take those really high quality molecules that come out of that process and again, characterize them in these complex disease-relevant cell-based assays. For us, that's a T-cell killing assay. We jump directly into the T-cell killing assay, multi-day assay, we get really rich dose-response curves from those, and that's the data that we're then using to train the models. I'll maybe just take a pause here and just say that's actually been a learning and an observation for us, that if you want to run multiple cycles, it's really important to gather data that is going to be comparable across different batches where you see quite high batch-to-batch variability. So in our case, having those full dose response curves across multiple different PBMC donors while you're doing the training and the data collection has actually been really key, rather than relying on maybe a faster single point readout. What I'm going to show you now is some of the molecules that came out of this process. So here we go. So what I'm showing you now are 3 of the really high-performing molecules that came out of this design, build, test, learn loop. And I'm really excited by these. So what you can see in dark red is the killing performance of these T-cell engagers against our disease model cell line. And what you're seeing in pink is the activity of these molecules against healthy cells. So again, those healthy cells expressing lower levels of TAA, the disease cell expressing higher levels of TAA, but there's only a 3-fold difference there in levels of TAA expression, and yet these molecules are still able to distinguish between them and really selectively just kill the disease cells. So rather than looking at that traditional shifting curve, what we've actually got here is something closer to that goal of achieving complete selectivity. And of course, this is tested across multiple different PBMC donors, as you would expect. Really excited by these molecules. They're now moving forward into animal model validation. Now, one thing that we have observed is we have got this really systematic process nailed now, and we've applied it across multiple different tumor-associated antigens to the similar effect. So what I'll do now is just for the sake of demonstrating this, show you another case study where we've done this across a completely different TAA. Okay, so in this case, this is a HER2 CD3 target, and again, in this case study on the left there, what you see in blue is a clinical benchmark, that's rudimotamab, and you can see the activity of rudimotamab against disease cells and healthy cells. In the healthy cell model, we took as a cell line with HER2 expression, with similar levels to cardiomyocytes, because cardiomyocytes have the highest level of HER2 expression in the human body. And again, you get that classic window there, that window of selectivity in rudimotamab with the activity on healthy disease cells. But again, using our process, we're able to come out with completely novel molecules, again, with something that looks quite similar to complete selectivity. So you very high activity, high efficacious killing against the disease cells, flat line against the healthy cell. And from a scientific perspective, I think one of the things that I personally find most interesting about this process is when you double-click into the underlying design of these molecules, you actually see molecules that you would never have designed using conventional rational-based methods. So what we have is the machine learning algorithm pulls us into areas of design space that we as human scientists may have never considered before. And I think this is, for me, a broader point about the applicability of machine learning for the design of therapeutic antibodies, which gives us the opportunity not just to create molecules that are more powerful in terms of their performance, but also molecules that we would never have found using conventional methods. So we are deploying this capability for our own internal pipeline for the development of both TCEs and ADCs, but we're also interested in deploying our capabilities across a range of different modalities. So I'll just end by thanking everybody for their time and their attention. I'm happy to take either questions now, or if you want to chat one-on-one afterwards, that's also great.
Very promising technology and a very great talk, so thanks for this. But my question is rather more from the biology side. So when you talk about the number of receptors in the healthy versus cancer cells, you assume that they are distributed homogeneously, but in many in vivo conditions, they are clustered in these islands, and then the density does not matter that much as you would think. So have you thought about this when you go in vivo or try more animal testings?
Yeah, it's a great question. So, and I would say not only that, but you've got the added challenge of what about the tumor heterogeneity as well? You know, what does the TAA expression look like across the different tumor cells, etc. Across these different challenges, you would hope with some of these modalities, you should obviously get bystander effects, which will help deal with some of those aspects of heterogeneity. But I think your question touches on a really important point to the application of machine learning for any of these applications in general, which is what is the biological relevance of the assay to which you're optimizing? And that's been the big, I guess, emphasis for us at LabGenius, which is to say you have to hit a certain throughput in order to be able to use the data for machine learning, and in our case, that's a few thousand data points. But then what's the biological relevance of that? So we're constantly pushing to increase the biological relevance of the assays that we conduct our process on, but it has to be high enough throughput to actually train a model. Now, of course, once you have these high-performing molecules, then, of course, we can do all of the follow-up assays, PDX assays, CDX, etc. that will hopefully surface some of those pieces. But it's a great point, and thank you for the question.
I have a question on how easy is it to generate these 700 data points that you showed in your test case. Could you, let's say, produce each of these T-cell engager in 4 milliliter turbo CHO, and was it sufficient to make a biological assay?
So we, for that second lead optimization stage, all of the production is done in-house. And then the process that we use there, and I should say, these are panels of 768 molecules at the moment, but they'll increase to 2,300 at the end of this year, just through pushing on the automation side. These are small-scale mammalian cell cultures where we typically get on the order of 30 micrograms of protein out of each of them, which is enough for us to run our in-house assays. Yeah.
Very nice lecture, thank you. So how many data points do you have to generate with to optimize your ML in the wet lab with your high throughput assays? Yeah.
So the type of, I should just say, people say machine learning, AI, etc. There are so many different types, and the specific type that we use, multi-objective Bayesian optimization, is really good when the amount of data you can generate is quite small. And that's actually really intentional for us, because what the approach does is you feed it data, and this is on the order of a few hundred data points, and then it says, here are the areas that I have a high degree of uncertainty over, here are the areas I have a low degree of uncertainty on, and then it will say, go away and produce data so that I can maximize my learnings. Yeah, correct. Yeah, exactly. Thank you.
Really great talk. Can you comment on the time frame or challenges of translating all these great technologies into a clinical setting?
Yeah, I mean, I'm obviously the CEO of the business, so I'll talk about it from a financing perspective, which is which is not insignificant. So, if you're looking for the most capital efficient way to progress a molecule into the clinic, then anyone would question, why are you building a platform? It's a massive capital investment. It's a huge time investment. The reason that we're doing it is because we think that with this approach, we'll be able to find molecules that have a higher probability of success when they do enter the clinic. So I would say the emphasis for us has been on build a platform, demonstrate that the platform can create higher quality molecules, and we should see the first of these molecules progressing into the clinic within the next sort of 18 to 24 months.
Okay, this will be the last question here.
Interesting technology. I was wondering, how do you initially pick the seeds to sample the design space? Because as you said, it's sometimes surprising what you find, so I guess it's hard to predict where to put your seeds as well.
Yeah, so, and this is where the multi-objective Bayesian optimization approach is so powerful. So our scientists will say, in this case, here are the 270,000 designs that we want to explore. And then in the first step, you try and uniformly sample the space. Now that's quite challenging because you've got a mixture of continuous variables, like say affinity, and discontinuous variables that are descriptive of your molecular design. But there are a variety of mathematical methods that you can use to try and then uniformly sample it. So you try and get that low resolution snapshot of the whole space, and then in the subsequent rounds, you're typically pulled into a few different attractor basins that can be quite distinct from one another. But what that buys you is a lot of diversity in that final panel of molecules. So you haven't entirely zoomed in on one area of high performance and just have lots of different small permutations around that. Often you can find these quite distinct areas of high performance.
Thank you again. With this, we can close